In our video today, we're going to talk about nucleic acids. And nucleic acids is maybe not a term that you've heard before, but it's an essential piece of all living things and sustains life. And this is for section IB A1.2, the new 2025 IB curriculum. For nucleic acids, maybe not the first thing that you think of is DNA, but they're actually one and the same, and they do very much go together. DNA you probably have heard of, and really DNA is a form of genetic material, and it's a tool that life uses to be able to store information. And it's really not that dissimilar from a book or binary code for computer programming, in which information is stored in a series of letters or ones and zeros for binary code, and then that information has some sort of meaning. For binary code, some sort of program is produced, and for books, then obviously a message can be read. And for genetic material, DNA or RNA, it's very, very similar. DNA stands for deoxyribonucleic acid, and it's a nucleic acid because it was discovered in the nucleus. RNA is another type of nucleic acid. There are large molecules that are made up of smaller pieces called nucleotides or nucleic acids. And so when nucleic acids and nucleotides are put together, they can make DNA or RNA. All living organisms have DNA, and we'll talk about a little bit later how that DNA varies from one species to the next, but all living things have DNA. So the components of a DNA nucleotide are really three parts. There's the sugar, deoxyribose, nitrogen base, which can be a number of different types, and the phosphate group. And so those three pieces put together make up a DNA nucleotide. RNA is pretty similar, has a different type of sugar that we'll take a look at a little bit later. The sugar has got 5 carbon atoms in it, and in our images here we can see both a molecular and a simple shaped drawing with triangles and rectangles and circles to represent our individual nucleotide. DNA and RNA are polymers of nucleic acids or nucleotides. And in order to put them together, covalent bonds are formed between the phosphate of one nucleotide and the pentose sugar of the next nucleotide. And this then creates what's called a phosphodiester bond. It's 2 different ester bonds put together, as we can see in the images here in the video. What this does is it creates alternating sequences of sugars and phosphates, and so DNA or RNA has kind of this backbone of sugar and phosphate, and it's this repeating pattern of sugar and phosphate, and so that backbone of both DNA and RNA is consistent. It's made of those 2 different parts, and what makes each of the molecules of DNA and RNA different then are the types of nitrogen bases that are attached to the sugars. The nitrogen base is always attached to the sugar, and this backbone helps to conserve the sequence of the bases of DNA or RNA. There are 4 types of DNA and RNA, and they each contain nitrogen, and thus they're called nitrogen bases, not surprisingly. Any of those nucleotides of DNA and RNA can be linked together in the backbone. And so that allows a sequence of DNA or RNA to be created that can vary from one individual to another and from one species to another. And it also allows or enforces the 2 branches within DNA, or the 2 chains or ladders of DNA, to be linked together. And so those 4 different types of nitrogen bases, one group are called purines, and these are nitrogen bases that have 2 rings of atoms as part of the molecule, and that includes adenine and guanine. And the opposite of that are the pyrimidines, and there's 3 different types: cytosine, thymine, and uracil, and each of those only have 1 ring of atoms within the molecule itself. And so within DNA, it's a structure of kind of like a ladder, where it has 2 backbones, as we talked about, and rungs in the ladder. And because one purine always binds with one pyrimidine, those rungs of the ladder are of equal length. And so that helps to ensure the stability of the DNA structure itself. RNA is a little bit different than DNA, and one of the biggest differences is that it's just a single chain. It doesn't have 2 backbones, it has just 1. So it's an unbranched polymer of nucleotides. And nucleotides are monomers. It's just like one piece that gets put together with more monomers to make something called a polymer. Mono means one, poly means many, and so if we put multiple monomers together, it makes a polymer. And RNA has nucleotides just like DNA, and so an individual nucleotide is a monomer, and as we put more and more of those together, 3 or more makes a polymer. And then in this case, the polymer that we're discussing is RNA. And those individual nucleotides can be put together. It's unlimited in how long that is, and it's formed by a condensation reaction. And so a condensation reaction takes place when water is removed. And in this case, it's the removal of an OH, an oxygen and a hydrogen from one molecule and a hydrogen from a second molecule. And the remaining oxygen forms a covalent bond with the carbon in the backbone. So the nitrogen and carbon in the bind together in the nitrogen base. Water is removed, and then those two molecules are bound together, as we see in the image here. And so then this creates a single unbranched polymer of nucleotides, and is formed by condensation reaction. So you maybe have heard the term or phrase double helix before. We've talked about DNA as being kind of the shape of a ladder. And that's, for both of these, the case because DNA is two strands. And so we call it the double helix as the shape twists. It forms kind of a spiral staircase with the steps of the ladder being the different rungs. And those rungs are made of the nitrogen bases that we just talked about, bound together. And the two strands are parallel to one another, but they run in opposite directions. So we call them anti-parallel, in which one strand ends with the phosphate group and the other ends with the deoxyribose sugar. And this is necessary because between the nitrogen bases, hydrogen bonds form, and that's what connects the two different sides together, is the hydrogen bases between the hydrogen bonds, between the nitrogen bases. And by the two strands being anti-parallel and going in opposite directions, that allows those hydrogen bonds to form with one another. So we've mentioned briefly that there are some differences between DNA and RNA, and now let's discuss those and identify those. Really, one of the main differences, as we discussed, is DNA is double-stranded and RNA is a single strand. That's a huge fundamental difference, and we'll talk more about RNA a little bit later on. One of the other large differences is the type of sugar. RNA uses a ribose sugar, whereas DNA uses deoxyribose, different types of sugars, as we can see in the images here. The DNA molecule uses the nitrogen base thymine, and RNA does not use thymine. Instead, it uses uracil. So DNA doesn't have U or uracil, whereas RNA doesn't have a thymine. And then the double and the single strand, as mentioned. So in order to ensure that the pairs match up, adenine and thymine pair up, and guanine and cytosine pair up within DNA, in order to ensure that they are complementary, pairing to one another, it really ties into the replication of the DNA molecule itself. And the DNA molecule, as being two strands, in order to replicate, it actually gets pulled apart, and those hydrogen bonds are broken between the two different strands. And each of those strands is used as a template for replication. And so complementary bases are paired with the templates. We'll discuss the actual DNA process a little bit later on in this first year of IB Bio. But for right now, it's just essentially the two strands get unwound and they act as a template, and new strands are added formed by adding new nucleotides one by one and linking them and creating new hydrogen bonds so that the bases are complementary. So if there's an adenine, then it's only going to be paired with a thymine, and a guanine with a cytosine, or vice versa. This ensures that two identical strands are created, and this process is called semi-conservative. And what this means essentially is that of the original DNA strand, each new DNA copy, so we have two copies that we end with, they possess one original template strand and one new strand. And we can see that in our picture here, semi-conservative. So one original strand is conserved and gets one new one. And that's the case for both of our two copies. And so semi-conservative is the phrase that's used to describe the replication of DNA. The diversity of DNA sequences is really essentially limitless. There's massive number of different combinations and sequences that are possible because of the ability to combine different nucleotides that contain different types of nitrogen bases, A, C, G, or T, in any amounts and in any combination. And this is ultimately what makes organisms different from one another within the same species and different species, is the length of that DNA sequence and the sequence itself, the A's, T's, G's, or C's. And so this provides a limitless capacity to store information. And so if we have just one base we have A, C, G, or T. If we have 2 bases, then we have 16 different possibilities. If we have 3 bases, 64, and so on and so on. And so while many species share very similar DNA sequences between different species, and members of the same species have even more similarities, there are still differences. And these differences then are what are responsible for creating different proteins. DNA ultimately acts as the template or the information to be able to make proteins. And proteins carry out all the functions of life and help to build life. And so DNA or RNA is coded by groups of 3 bases called a codon. And those codons are found in RNA. As we'll see when we get to DNA replication and protein synthesis, the DNA is turned into messenger RNA for many organisms, and that RNA then is used to make proteins. And there's 64 different types of codons, most specific particular to amino acids. One is a start codon, and there are 3 different stop codons. So it's kind of like an on and an off switch. So there's 1 start codon and 3 stop codons, and then the rest make up different types of amino acids. And you probably have heard of some of these different types of amino acids, and we as humans acquire them by eating food, by what we eat. All living organisms and viruses use the code to be able to carry out their functions of life. It's called the universal genetic code because it's used by all species. So DNA is universal because all living things use it. And over time, that code has changed in different ways in different species. But because all forms of life use that same code, it suggests a common ancestry. And so this really serves as evidence of a common ancestor, and that also because all of the species use DNA as a means to store genetic information and to store information to be able to control life. It suggests all life shares some form of common ancestor. The direction of DNA and RNA is similar. Both have nucleotides that are linked in the same manner, in which the phosphate group of one nucleotide is linked to the sugar of the next nucleotide using an ester bond. And so we have those phosphodiester bonds that we previously talked about. All the nucleotides in one strand are orientated the same way. In DNA, those 2 strands are anti-parallel. And again, this allows the hydrogen bonds to form between the different nitrogen bases. And so although RNA only has 1 single strand, it's still orientated in the same way of the sugar is connected to the phosphate between 2 different nitrogen bases. When we look at the image here, we can see that the 2 strands of DNA are opposite directions, and that's helped signified by we can see the numbers with little asterisks next to them, the 5' on one side and the 3' on the other, and then on the opposite strand we see 3' and 5'. And so that's indicating the different directions of those 2 molecules, and that's really essential to our next step and next process of enzymes that help to facilitate replication or the making of proteins through transcription and translation. It's essential to have those anti-parallel directions. And new nucleotides during DNA replication are always added at one particular end. That's the 3' end of the molecule. Enzymes move towards the 3' end. So it's thus 5' to 3' for replication, for transcription, translation. All of these enzymes that help to coordinate these mechanisms, these steps, are dependent on that. During replication, both strands are replicated at the same time in order to be able to create 2 identical copies of the DNA. And so one strand is going to move in the direction of the overall process of replication, and the other moves in the opposite direction. So this makes a little bit of complications when DNA is replicated, not in terms of the process, but just in terms of our understanding of the process, as we'll see a little bit later on. Transcription, only 1 DNA strand is used as a template. That 1 strand then is what makes RNA that's used to make proteins, and how those proteins actually get made are in the process of translation, and that's then the linking of amino acids together to make a protein. And that process also uses enzymes that move towards the 3'. While for the majority of the time DNA is actually this long wispy structure that's not really condensed, there are times when DNA condenses down to be able to be moved around. And this happens during cell replication when the cell is actually dividing to split into two, and it forms probably what you've maybe heard of as chromosomes. And so during this process of the DNA forming into chromosomes, there are structures that help to stabilize the DNA and can also influence transcription. We'll talk a little bit more later on in the year. And these structures are called nucleosomes. And a nucleosome is DNA that's stabilized by some proteins. And so there's DNA that wraps twice around this bead or chunk of proteins, and there are 8 histone proteins that are together kind of in a little capsule that the DNA wraps around 2 times. And this helps, again, to stabilize the DNA within the chromosome and can also influence the rate of transcription when proteins are being made. While it's easy for us today to say that we know DNA is the source of genetic information and stores all of our information for us as individuals as well as our species, that's not always been the case. And one of the coolest things about science is how new information develops all of the time. And back in the 1940s is when we as a scientific community first began to understand that DNA is the source of genetic material. And 2 scientists helped to demonstrate this with a particular experiment that they conducted. Alfred Hershey and Martha Chase conducted an experiment that helped to establish that DNA is the genetic material and provided that evidence. And so what they did in their experiment is they, using the understanding that DNA contains phosphorus, but not sulfur, and proteins can contain sulfur, but they don't contain phosphorus. So there's some fundamental differences between these two because it wasn't really known whether it was DNA or proteins that was the source of genetic material. So DNA has phosphorus, protein has sulfur. And so what Hershey and Chase did is they took a virus and they added radioactive sulfur to some of these T2 viruses, and they added radioactive phosphorus to other viruses, so 2 separate groups of viruses. And radioactive allows it to be detected, and so they could track where was this sulfur and where was this phosphorus. And so they took the viruses that had the sulfur and the phosphorus, the radioactive, and put them into some bacteria. Viruses infect bacteria, and so the viruses then would release their material into the bacteria. And what they discovered after using a blender to separate the genetic components and non-genetic components from the virus, they noticed in the later generations of bacteria that there was radioactive phosphorus present, but no radioactive sulfur. And so if we remember back, the radioactive sulfur was attached to the protein and the radioactive phosphorus to the DNA. And so what this showed, because those bacteria persisted over a couple of generations, they replicated, and over a couple of generations, they found that generations later in the bacteria, the radioactive phosphorus being present indicated that it had to have come from that first generation. So it had to have been passed down from parent to offspring over the course of those generations. With no radioactive sulfur being present, that means and indicated that it wasn't being passed down from one generation to the next. So this helped to provide evidence that the DNA is actually the genetic material rather than proteins. And we know that today now thanks to Hershey and Chase and their experiment. A second experiment helped to confirm the basis of Watson and Crick's double helix model. Originally, it was thought that DNA's structure was a tetranucleotide structure with a repetition of 4 bases in the sequence. And so this would have suggested that there would have been equal amounts of all 4 of the bases, adenine, guanine, cytosine, and thymine. And this was originally suggested by Phoebus Levine, proposed this tetranucleotide structure. Another scientist later on came along, Chargaff, that proposed, and after doing some analysis, that was not a tetranucleotide, but that the arrangement was just 2 nucleotides, so adenine and thymine together, and guanine and cytosine. And he discovered this by analyzing samples of DNA from a wide range of species to find their nucleotide composition. And in any species, the amount of guanine and cytosine should be equal because they pair together, and adenine and thymine pair together, and so those amounts should also be equal. And because after calculating the amount of each of those, he found that the amount of guanine and cytosine was equal and adenine and thymine was equal. It helped to confirm that there was not a tetranucleotide structure, but rather just two nucleotides put together.